These are going to be slow responses, but it will be highly selective, so very different from innate defenses. However, they are going to work together. And so with specificity, so these will have specific targets where these will be determined by different antigens. So an antigen will be any molecule that host doesn't recognize as itself within the body. And so we have a systemic response where it's not restricted to just an initial site of infection or invasion. Our adaptive defenses also have something called memory, where after initial exposure and activation, there will be a more rapid and a more vigorous response that is going to be made to that certain pathogen. And so this is what's termed as our secondary response, where we'll have a shorter time for the immune response. We have two different types of immunity with adaptive defenses. So the first one is with humoral immunity, and this has to do with our B cells. So let me write that down. And this is where we have a wide diversity of targets where antibodies are going to be free floating in the body and will be continuously created. The cell-mediated immunity, this is due to our T cells. And so this is where the lymphocytes are going to directly attack specific invaders. And so they're either going to cause those invaders to swell or lysis, or to indirectly attack them by starting the inflammation response. And so here we'll also have activation of other lymphocytes and macrophages to aid in this process, where this must be close to or be touching the victim cell though. So that's going to be a little bit different, whereas with the humoral immunity, that's going to be within the entire bloodstream. However, with the cell-mediated immunity, it has to be close to that cell. Regarding the humoral immunity, what the body is looking for are different antigens. And so antigens are going to be something that's foreign to the blood or other bodily fluid and will initiate a immune response. And so this includes things like proteins, nucleic acids, lipids, polysaccharides, and they also have the ability to react with other lymphocytes and activate them, as well as different antibodies. However, in regards to antigens, they have different recognition sites called epitopes, and this is where antibodies will bind to antigen, and then that will initiate an immune response. And so we'll have the humoral working with the antigen, and can work with a cell-mediated immunity response as well. So the two different types of lymphocytes that are involved are going to be, again, the T cells and the B cells. So again, the T cells, these are going to be with cell-mediated immunity. And then with the B cells, if you can remember antibody, which I'm just going to abbreviate AB, mediated responses. And so with our T cells, these are going to be produced within the thymus. And then the big thing is that they don't secrete antibodies. Okay. Now, B cells, on the other hand, these are going to be in mature bone marrow. And so part of their names for these cells are where they are produced. And so these are going to be responsible for bacterial infection and some viruses. And these are going, again, to be related to antibody production and secretion. A little more detail about the different types of cells for the immune system. So the T cells, again, within the thymus, what they do is they attack host cells and become infected with a virus or fungus. Also, examples are transplanted human cells and cancer cells, anything that doesn't recognize it as itself. And so these must be continuously produced to provide immunity. However, big thing is that they do not produce antibodies, okay? They must be in close proximity, so they need to be very close or almost touching the victim cell in order to destroy it, and so these help to manage the immune response. B cells, on the other hand, are going to be sorted in the bone marrow, again, BB, And so these are for bacterial and some viral infections, but the big thing is here that they secrete antibodies, so BB. And what happens here is that after many cell divisions, these B cells can become memory cells, which are important for active immunity, or plasma cells, which produce about 2,000 antibodies per second. And so they're responsible then for initiating that immune response, and those T cells then are responsible for maintaining that immune response. So with maturation of both B cells and T cells, they eventually will become immunocompetent, where they're going to recognize specific antigens. However, if you remember a couple of slides ago, we talked about, though, that antigens have different epitopes for which antibodies will bind. So not all antibodies that are produced will recognize a certain epitope. And so when we have the receptors on one cell that are identical, though, the immunity will depend on genetic coding for appropriate receptors, and which all will depend then on the epitopes of those antigens for which will generate the immune response. So if we have an antigen that our body has already recognized to a certain epitope, an antibody has been produced, and so the next time we're exposed to it, we'll have a quicker reaction rather than trying to make a new antibody for that certain epitope of that antigen. When we look at this image, let's begin by going over, so from stem cells, we have the two different types of lymphocytes. We've got B lymphocytes and T lymphocytes, where again, the differences will be for the T lymphocytes, these are going to be cell-mediated responses, versus the B lymphocytes, these are going to be antibody-mediated responses, okay? And so let's begin by looking at the B lymphocytes. And so once B lymphocytes are produced, what they can do is that they can clone one another. And so this is when we have an antibody plus antigen binding, and that's when that occurs, okay? And so we have two different types that are generated. We'll have memory cells that are produced, or plasma cells. So the memory cells will be long-lived as compared to the plasma cells, which will be short-lived. All right, and so this is going with the memory cells, then we'll have a shorter immune response as compared to the plasma cells, where we'll have the generation then of antibodies. So why this is short-lived is because we have antibodies produced. However, the memory cells then, when they're exposed to that same antigen, are going to produce more and more of those antibodies, and so that takes a shorter amount of time. Okay, so then difference with now T lymphocytes, so there are different types that can be produced. So we have helper T lymphocytes, which can either turn into and activate B lymphocytes, or can help initiate the C cells, or cytotoxic killer T lymphocytes. And so again, these are going to need to be in contact with their victim cell in order for them to generate a response. So problems that can occur, though, with these killer T cells is like with autoimmune diseases, where for whatever reason there is some discrepancy in terms of recognition of self, and the self cells that wouldn't kill themselves start killing themselves, and then start the apoptotic pathway. And so like multiple sclerosis would be an example of this. With our humoral immunity, which again, with our antibodies, so these, there are different ways we can acquire them. Naturally would be one example, and so this is natural exposure to the antigen, and so this is like due to infection. Also passed down maternally, we'll get that as well. Artificially acquired immunity requires vaccines, and these are either dead or attenuated or fragmented pathogens that are injected to elicit some type of response. And so we'll have a immunity without the disease, and so this is the primary response, where the primary response takes about 5 to 10 days before measurable amounts of antibodies appear in the blood. A secondary response, these are a lot quicker, and so these might only take 1 to 2 days for the antibodies to be or to appear in the blood. Some shortcomings, though, that occur with artificially acquired immunity include like adverse reactions. So like sometimes if you have the flu shot, you might get sick anyways, and so the immunity has a poor memory, or there's less cell-mediated parts to your immune system that need to take time in order to attack that artificially acquired pathogen. So when we look at antibodies, so they have this typical Y shape to them, okay? And so these are due to different types of protein chains that we see. However, with all antibody molecules, we'll always have some type of antigen binding site. And so this is based on the polypeptide sequence of proteins that we have. And with these Y-shaped proteins, also we'll have 2 long, heavy chains. So here's one, and then here to be the second, and then also by 2 short, light chains. Okay, the bottom here is going to have the same structure with all antibodies, and it's up here at the top of the Y is what will determine what antigen that antibody will bind to. So to give you an idea as to how many antibodies we have, so everyone has 10 to the 20th power of antibody molecules circulating within their bloodstream. And so there are a few million different specificities, and that should be an antibody for antigen that you might encounter. However, this is not true because of the amount of epitopes that the antibody can recognize. So when we have the binding of the antigen to the antibody, that's what's going to trigger then our immune response. We have different classes of antibodies, which will depend on when these enter the blood. That's what a big difference is to them, okay? And so Ig is going to stand for, so let me write this down, Ig equals immunoglobin, okay? So we'll have IgG, so it's immunoglobin G, and this is the most abundant circulating immunoglobin, and it has the structure to it. And this is responsible for the primary and secondary responses that we see, and this is what's able to cross the placenta when we are in utero. Immunoglobin A, this is responsible for preventing antigen penetration of different membranes, and so structure is a little bit different than immunoglobin G. Helps to protect the body from inhaled or ingested microbes. Immunoglobin D, this is important for beta or B cell activation, and so we find this on B cell surfaces. So this is what a typical antibody, what it looks like. And so this will bind antigens in the extracellular fluid and initiate B cell activation. Immunoglobin M, this is what that structure looks like. This is the first class of antibodies secreted after an antigen appears, and it's only on the B cell surface. And this is primarily responsible for plasma cell response, circulating antibodies, and then also agglutination. So we saw that with our blood, where we had incompatible blood typing. And then immunoglobin E, these you might be more familiar with because these are what are associated with allergies. And so these will bind to mast cells and basophils, and then they release histamine, and induce inflammation, and found mostly in mucosa of the respiratory and GI tract. And so they have this pentamer shape to them. Some common traits for all antibody targets and functions include that they have this complement fixation. All right, and so this is where we have lysis, or the swelling and bursting of cells and bacteria, and this is due to an increase in cell permeability. Okay, and so that allows for, that increase in permeability allows for those lymphocytes to get in, and then those antibodies then will start attacking. All right, so the next one is neutralization, and so this is where we have the blocking of specific sites on the virus or the toxin, and it inhibits then binding to tissues. So the purpose of this then is for inactivation, okay, of that foreign invader to our host cells within our body. All right, the next one, agglutination. Again, you're familiar with this, doing this in lab and blood typing. And this is where cross-links occur in the immune complex. And so what happens is that inactivity occurs. So inactivity. And this happens within the bloodstream. So the primary antibodies responsible for this are going to be immunoglobin M and immunoglobin A. All right, the next function has precipitation. And so with this, we have soluble molecules, which are typically toxins, that will be cross-linked to make them insoluble so that they cannot penetrate into our cells. And then the last one will have inflammation and phagocytosis. And so with phagocytosis, this is due to opsonization. And again, this starts the coding of the bacteria so that we can start recognizing it as a foreign invader. And so that will help to have our immune response begin, because now we're starting to recognize it as foreign molecules. Some immunodeficiencies and dysfunctions that can occur with our immune system. There's two different types. So the first one are congenital or genetic, and so these are due to inborn errors, and some of them are just as simple as one codon could be misread, and or it could be multiple. So it could be on multiple chromosomes, or it could be just on one. Acquired, so these could be due to several different reasons. So the first one we're looking at with different drugs. So immunosuppressive drugs or cancer drugs can decrease and inhibit the immune system, mostly attacking the bone marrow. So with our B cells. Also cancers, these are viewed as failure of the immune surveillance. And so instead of killing cells, we usually will have an increase in production of cells. And then we'll have another example is Hodgkin's disease. So this is specifically within the lymph node and within our lymph node cancer. And then HIV and AIDS, this is where the helper cells are being killed. And so there's a decrease in number of lymphocytes. With autoimmune diseases, this is where the production of antibodies and helper T cells are against itself, and so it's recognizing itself as a foreign invader. So common examples of this include MS, multiple sclerosis, which will attack the myelin, the white matter of the nervous system. Graves' disease will attack the thyroid, and so will affect metabolism. Type 1 diabetes mellitus, so this will attack beta cells of the pancreas, and type 1 was that juvenile diabetes, commonly referred to as that. Rheumatoid arthritis, so this is where the joints are being attacked, and it's very painful because you're not able to move your joints as you used to. And then glomerular nephritis, this is where we have renal function that's being attacked, and so ultimately can cause kidney failure as well. So some of the reasons why this self-tolerance or this recognition of self fails is that the antigens might not normally circulate in the blood due to when it's exposed in the immune system. Another example is when self-antigen is combining with foreign haptens, which are antibody-producing proteins. And so this is when, for example, low platelet count with pharmacological drugs like aspirin. Another example would be with antibodies attacking other antibodies, and that's like the case with rheumatoid arthritis. And then lastly, antibodies produce foreign antigens, and so like strep throat then can affect the heart and damage it. So it has multiple targets, not just one. So some of the mechanisms and functions of the decreased immune system would be failure of the lymphocyte to program correctly, and that could be due to like stem cells that are being affected at that level. Also, there could be new self-antigens that are being recognized, and so like with gene mutation, this is typically caused by carcinogens. And so what they start to do is produce tumor-specific antigens instead of non-tumorous antigens. Also, different structural changes can occur within the antigens where the different epitopes then will create different sources of infections. Another one are going to be these different proteins of haptens that will affect them as well. And also foreign antigens that are very similar in resemblance might result in cross-reactivity, and so that would decrease the immune response and efficiency of the antibodies. I'm sure you've heard of different allergies or hypersensitivities, and this is where the body responds to harmless substances as if they were threats. And so like wheat is one, pollen is another, and so an allergen will be when an antigen of an allergic response occurs, and commonly it's due to an increase in histamine release.